In this final installation of the blood typing lab, I want to show you how to try to determine the genotype of a blood type from the phenotype. You should have already watched the second video of this three-part series in which I actually did blood typing using one of the synthetic bloods, not sure which, to figure out what the blood type is. So the blood typing slide looked something like this. And we had A, RH, and B, like this. And so here's what we saw. In the RH area, we saw no clumping whatsoever. It was very smooth. I can't draw these lines any thicker, unfortunately. At least I don't think I can. Can I? No. Doesn't really work. So no clumping. In the A section, we did see clumps occur, large ones. That was pretty easy to see, but we saw clumps. Also called agglutination. And then in the B, we saw very, very small clumps, but they were definitely clumping. And any clump is a clump. But it was all throughout the thing. It was just really, really small. So let's see what this means. We had, in the A section, we had agglutination, right? So that means that the A antigens were reacting. Let's rephrase this. The A antibodies were reacting with the A antigens. So that means that A antigens had to be present in this blood. So this person has at least an A in their blood type. We don't know what else yet. So let's take a look at B. In B, we saw clumping, right? So that means that the B antibodies in the serum that we put in were reacting with the B antigens. So there must have been B antigens present. Now, there is no clumping found in the RH factor. So that means if there's no clumping, no clumping means no antigens to interact with the antibodies. Cram it in there before we get to the camera picture. So this individual has to be AB negative. That's their blood type. Because we saw clumping in A, we saw clumping in B, but no clumping in the RH factor. So they are AB negative. So let's see what their blood types actually are and the genotype, because this is their phenotype, the AB negative. Let's figure out their genotype. So if we have an AB negative individual, our phenotype is this. The genotype, though, are the actual alleles that they carry. Now we know that an AB individual has to have IA on one chromosome, and they have to have IB on the other chromosome. Now, remember that these are codominant, so they express at the same time. We can have an AB individual, so that's what this is. Now we can also look at the RH factor. The RH factor states that there is no clumping there, so this individual must be RH negative and RH negative. on their chromosomes. Cool, right? Piece of cake. So what if, though, this individual instead were AB positive? Well, we could have IA, have to have IA, have to have IB for those to express together on the chromosomes. But there are two options. Do you remember when we said that if you know the recessive phenotype, you know the genotype? Well, that's the case in this first example. We know that it's negative, and that's the recessive phenotype, so we know its genotype is Rh negative, Rh negative. However, we have a positive, which is a dominant phenotype. So we can either be IA, IB, Rh positive, Rh positive. Well, let's do it as if we're doing the chromosomes. Or they could be IA, IB, Rh positive, Rh negative. They could be heterozygous because it still expresses the positive phenotype. So there are a couple of questions on the lab handout that involve this, and I'm going to let you do those on your own and with your group during lab time. But I hope that that helps answer the question on how to figure out the genotype of blood type from the phenotype. All right, that's it for these videos. Thanks so much. Have a good one.